Hi, welcome back. In this section, I just want to talk some terminology before we try to put things to work. And the first part, we're going to talk about various types of cytokines. We'll come back and revisit that later. Also, something called MHC, which is major histocompatibility complex, and then CD, clusters of differentiation. So, cytokines are protein messengers, and we have hundreds of these. And basically, what they're going to do is they regulate the cell's division and their function in immune processes. So they determine if the cell is going to undergo mitosis. So instead of having one cell fighting it, you're going to have two or an entire clone going on. And they are produced by many different types of cells, the majority of which are probably immune cells. So cells you've heard about before: monocytes, macrophages, lymphocytes, granulocytes, mast cells, even endothelial cells in your blood vessels, and fibroblasts in the tissue. So with cytokines can be released from these immune cells. It also can be released when, for instance, there's a site of injury and some tissue is damaged. And what these do is they act as molecules for some signals for things to happen either between cells or within a cell. And so most of the action, especially in the immune system, revolves around the area where they are produced, as in they're going to be autocrine or paracrine, either acting on the cell that's producing it or acting it on its nearby neighbor cells. However, there are some that will travel and act in an endocrine manner. We're not going to be talking about those. So how do they do? Well, basically, or how do they do what they do? Basically, they're linking different cells together. They're how cells are going to talk to each other. And oftentimes, one cytokine that is released stimulates another cell, which releases another cytokine, et cetera, and you have this whole cascade that is induced. So if we look at the picture of what is there, it's showing that this cell, which was activated, and it's a helper T cell, as you'll learn, secretes a cytokine. In this case, they are referring to interferon gamma, which has an action on macrophages. And one of the actions is it causes production of an interleukin, in this case, interleukin 12, which we're really not focusing on. And interleukin 12 actually goes back and activates, works on activated helper T cells, the cell that started this whole cascade. And now this activated T cell will not only secrete interferon gamma, but also tumor necrosis factor and another interleukin called interleukin 2, as well as other cytokines. So we're only going to be learning about specific cytokines because, as I said, there are a plethora of them. Now, the other thing that's interesting to remember about cytokines is there's a lot of redundancy. In other words, there are multiple different cytokines that can do similar functions. So for instance, from that activated helper T cell is showing at least 3 different types of interleukin, which have an effect, in this case, on a B cell. So nothing on this page as far as specific actions of cytokines is anything you need to know. If you need to know the action of a cytokine, it's because I've covered it later. So here are some examples of cytokines that we are going to be talking about. Interleukin 1, as well as tumor necrosis factor alpha, and interleukin 6, which we're not going to be talking about. These are all cytokines that are released by a cell that's called an antigen presenting cell, which you'll learn about when you learn about cell mediated immunity. And the target of these cytokines are the helper T cells, as far as we are concerned. And when these cytokines act on the target T cells, it causes them to create a lot of receptors for interleukin 2, as well as going back and stimulating an inflammatory response. Now, interleukin 2 actually can come from most of our immune cells, and one of its targets is the helper T cells. And so when it binds to a helper T cell, as well as also binds to natural killer cells and B cells, but when it binds to these cells, it stimulates their proliferation. So here's what we have interesting. Just in these first 2 lines, we had interleukin 1 making helper T cells make more interleukin 2 receptors, And then these same cells that produce interleukin-1 can also produce interleukin-2. And now we have a bunch of interleukin-2 receptors on the helper T cell. So we have a lot of it telling that helper T cell to make clones of helper T cells. So that's really kind of a cool effect. As far as interferons, we talked previously about interferon being produced when we had a virus infecting any kind of a cell to produce antiviral proteins, and that was one of our non-specific responses. Now we actually have a different type of interferon that comes from activated helper T cells as well as natural killer cells, which works on natural killer cells in an autocrine kind of a manner, as well as on nearby macrophages. And what those cells end up doing is releasing chemicals which kill the cell that started this whole process. Chemokines, we talked previously about chemokines, a whole bunch of different kind of chemokines involved. We've talked about histamine previously. We've talked about helper. We've talked about a lot of different things. Anytime we have damage or release chemokines, some of them are attracting other cells. That is a biggie. It's like the little cookie trail, so they know where to go do their work. And colony stimulating factors, we're not going to be talking about them in this class. So it's beyond the scope. So that's it for the cytokines. So the next concept is MHC, which stands for major histocompatibility complex. And what this is, it's found on the surface of all of your cells. Unfortunately, has another name called HLA, or human leukocyte antigens. And when we're talking normal immunity, we tend to use MHC term. When we're talking about transplants, we tend to use the HLA term. So go figure, but they're exactly the same. It's kind of like the same as immunoglobulin versus antibody. So all these markers on the surface of your cells are what enables your body to recognize, hey, this is part of me. Yes, I appreciate you. Thank you for doing all your hard work. I'm not going to attack you. Now, if another cell comes by, say, for instance, you got a tissue transplant, it would have different MHC markers, and your body's immune system would go, hey, you don't have the right markers. You're not part of me, and I am going to attack and destroy you. That stimulates an immune response. And so if it was due to a transplant, you would reject that tissue. So when we look at MHC, we actually have two different types of molecules, okay? And this is important for T cell mediated immunity. So our cell mediated immunity, because T cells will only recognize an antigen if it is attached to the MHC2 marker. So there's two types of molecules. Class 1 MHC are on the surface of every cell in your body that has a nucleus. If it's got a nucleus, it's got an MHC1. So that's good. Class 2 MHC are on the surface of some of your immune cells. So your macrophages, your B cells, your T cells, and antigen presenting cells. So the way this is going to work is if one of those immune cells has an antigen bound to the class 2 MHC molecule, then the T cell can recognize the antigen. If it is not bound to this class 2 MHC, the T cell will go, antigen? What antigen? I don't see it. Do you see anything? Did you see, SARS-CoV-2? No, I don't see it. It's just a bad rumor. It's going away with the heat of summer, I guess. All right, so that's MHC. The last term is CD, which stands for cluster of differentiation. And so on one of your chromosomes, there's a whole bunch of gene markers. And basically, these are just codes for surface proteins. And the only reason why I want to talk about these is because we're going to talk about two different types of T cells, which are called helper T cells and cytotoxic T cells. But oftentimes when you're reading about them, they're called CD4 cells and CD8 cells. And so basically, CD4 cells are those that contain that protein, which was originally called CD4. and a different glycoprotein, which was called CD8. Those are on your CD8 cells, okay? And what both of these are molecules which enable both of these T cell subsets, the helper T cells and the cytotoxic C cells, to bind to other cells, because that's how they are going to work. They don't have an antibody attaching to a T cell, which then can bind to another cell. These are the molecules that allow them to bind to the other cells. So with that, I am done. I hope you understand all this terminology. Now go have fun and learn all about T cell mediated immunity and its effects on B cell mediated immunity. And I will see you in our next class. Thanks for all your work.